Today I'd like to talk about the brain. Now, in particular the brain, protein synthesis, and long-term memory. Oh, we need this. Okay, now, all of you know by now that the brain has an enormous amount of electrical activity. And this electrical activity is located at the membrane and the synapse. But the rest of the brain, you have no real concept of what it's doing. If you think about it for a second and take our archetypical neuron with its synapse, you realize now you have a pretty good understanding of what's happening only along its surface in the conduction of ions, impulses, movement of ions in and out of the membrane, and here at the synapse where neurotransmitters is released. But you have no idea what anything else in the entire cell is doing. The exoplasm, the pericardial region with the nucleus. You don't have any idea what's happening in the cytoplasm in the dendrites. Now, so probably you have a concept that the cytoplasm and the nucleus is all kind of a passive supportive structure that is supporting the functions of maintaining the plasma membrane and the neurotransmitter concentrations. This, in fact, is incorrect. The brain is unique and well known for its enormous amount of electrical activity, unlike any other organ in the body, or to an extent unlike any other organ in the body. But it's also very remarkable for an enormous amount of protein synthesis. As a matter of fact, the brain, along with the liver, has got the highest amount of protein synthesis of any organ in the body. Now this is a remarkable fact. 1/3 of all the blood that's pumped from the heart goes to the brain. In other words, an enormous amount of metabolic activity is sustained in the brain. An enormous amount of blood is going to this area to maintain it. Commensurate with this large blood supply is an enormous supply of nutrients essential to maintain this high degree of metabolism. A lot of oxygen goes, a lot of the amino acids, a lot of the basic RNA parts and DNA parts are going to the brain all the time, and CO2 is removed. So it appears that there's a lot going on that isn't really related to the electrical phenomenon. So what is it doing? That is the question that we are addressing to ourselves. It's true that we have to create neurotransmitter. It's true that we have to replace membrane ion channels and sodium pumps to maintain the ion gradient. But this is a relatively small amount of energy requirements that needs to be devoted to this one thing. So, for example, it is known now that a fairly large percentage of the brain protein turns over in 14 hours. Now that is, roughly 5% of this protein is synthesized, that is, there's a DNA messenger, the DNA template, and then there's an RNA messenger which goes out in the cytoplasm and is created protein. This protein is transported somewhere, is used, and 14 hours later it's degraded and it's gone. So every 14 hours, roughly 5% of its protein is used and turned over. This is a half-life. Now the other 95% doesn't fare much better. It lasts about 8 days. And again, in 8 days, you'll have a turnover of protein. Protein that you see at day 1 will be around for 8 days, and then half of it that's been created at day 1 will be gone in 8 days. So you could say roughly every 16 days, or every month, the brain is recreating its protein constituency twice. Now this is phenomenal. This is just, it's enormous. And well, the first question that comes to my mind is, with this kind of turnover continuously, a dynamic turnover where nothing is retained for very long, at least in the form of protein, and it's true for other constituents too, then how can anything like a long-term memory be created and stored? I could see how it could last for about 8 days or 16 days or even a month. But when the same proteins aren't even here after 13 days, then how is this long-term memory being stored? Okay. And the other thing is, why? Why is there so much? Why does the brain need such an enormous amount of metabolic activity to sustain its function? Especially when we know there's electrical activity that does not require this. Okay, the answer to these questions aren't simple. In fact, we just don't know. But we have some ideas, and I'll begin to present some data which supports a basic idea of protein involvement in long-term memory. And I'll try to give a reasonable explanation for why it turns over so rapidly. Question?
Yeah, by protein turnover, you mean also the protein blood in the membrane protein makes up the cell that also promotes?
All protein I'm referring to. I'm not distinguishing between membrane protein and cytoplasmic protein. Okay. This 14-hour protein that turns over very rapidly is located generally, is transported to the synaptic region. Okay. In 1963, Flexner, and Steller. This was not a very good year for science, by the way, but it wasn't bad. Discovered that if they injected pyromycin into the peritoneal cavity of mice, okay, pyromycin, this is called an IP injection. It means the peritoneal cavity is basically a visceral cavity. And inject it into the viscera and it's picked up by the bloodstream and circulated throughout the entire body. So it perfuses the entire body and it passes the blood-brain barrier and enters the brain. Now they found that injections of pyromycin, which is an antibiotic, and as Rick pointed out the other day, antibiotics are known to inhibit protein synthesis. So pyromycin comes in and at a 90% or better protein inhibition level, these mice did not incorporate and retain long-term memories of a task. What they found was that the mice would learn normally, they would acquire the task, and they would retain it for a short period of time, about an hour, in complete normalness. But 24 hours later, when they were tested for retention, they had no, or they had very poor retention of the tasks that they were trained in doing. But this was a focal point, a crucial point, which indicates that this protein synthesis is somehow involved with memory storage. Okay, so now people became fairly interested in the kinds of proteins that are found in the brain. And they were interested in perhaps maybe brain-unique proteins. And so looking around and using a number of different techniques, they found a number of proteins which seemed to occur nowhere else except in the brain. One protein, in 1965, Moore discovered and described protein S100. This protein was named because it dissolved in a saturated solution of ammonium sulfate, which is how he extracted it. And he characterized it as being a small, highly acidic protein. And when I say acidic, I mean it has a lot of amino acids that have acid attachments to them. It's a highly acidic protein. It has a lot of acidic amino acids in its constituency. Does everybody know what an amino acid is? Does anybody not know? Okay, well, just real briefly, just for everyone's benefit, a protein is essentially a string of amino acids. One strand, no branches. And that these amino acids are linked together, and that depending on the kinds of amino acids used, this protein will assume a particular configuration, and that configuration will confer upon it a particular function. When it's created in a strand, and when it's released, it curls up generally in some kind of globular conformation. Now, these amino acids can be basic, they can be acidic, or they can be neutral. Okay, so this had a lot of acidic amino acids. He found out that approximately 0.1% of the brain protein is this S100 protein. 0.1%. Okay, now, a little bit later, in 1966, Holger Dane discovered that this protein was primarily a glial protein. Okay, Holger is very important in this area of research. Swedish. Okay, now, so Holger Dane was pretty interested in the proteins, and in particular this S100 protein. So he developed a new technique where he took, under a microscope, would dissect out single cells of nervous tissue. This is a phenomenally difficult procedure because it's just so small and they're so fragile. So he'd sit there with microforceps and micro tweezers, and he'd tease out cells one at a time. And this way he could separate out glial from non-glial or nervous tissue. And this was an enormous step forward because in every study that you make where you just homogenize whole brain, you have vasculature from the blood, you've got blood cells, you've got blood plasma, you've got all kinds of glial tissue that's going to interfere and cause noise and perhaps cover up any kind of differences that you're going to look for that are happening inside nerves. So he sat down and developed this procedure by which he could tease out one cell at a time. And he'd sit down and he'd tease out hundreds and thousands of each cell. Enormous amount of work. So he would do this. Now, I'm going to describe one. He did a number of studies, and one of them, which I think is most relevant to our study of proteins and long-term memory, is one where he did a study on the handedness transfer of rats. Now, rats, just like human beings, have a handed preference. Most people are right-handed, and they tend to use the right hand for tasks. He discovered that 92% of rats had a distinct handedness preference. That is, they either used the right or the left paw in preference over 50% of the time, 75%, a rather arbitrary definition. So he created a task where first he would measure their handedness preference by putting in a cage a small cylinder that was partially filled with small protein pellets. So the rat would have to come along and dip his hand in and pull these pellets out one at a time. So he trained them to do this. And he'd observe which hand they used. And then the task became now to reverse handedness. If they were right-handed, he would move the cylinder in a position where it's up against the wall and he couldn't use the right hand to get in and get it out. So he'd have to start using the left hand. And in the beginning, there was a disadvantage, and they found that you could get fewer pellets out, but that after a number of trials, that this would improve over a period of days. So what he did is he plotted out the handedness transfer in terms of number of trials, and he gave 2 trials a day. For 25 minutes, and he recorded the number of pellets that a rat could pull out. And on an average, with a large number of rats, it looks something like this. That on day 1, after they had been forced to use the other hand, they can get about 50 pellets. And that it tends to increase linearly with each trial up till about day 8. And it gets to almost about 150 pellets a day, and then goes down a little bit, and then back up.
Drops to day 10, and then goes back up to day 10.
So roughly like that. There's a little dip, and it goes up by day 15. It's back up at a maximum that's the equivalent to using their original band of preference. So once this thing is trained, though, he finds that they continue using their less preferred hand after they've been trained. Now, he was interested in what effects his training had. And, okay, the beautiful thing about this manipulation is that he didn't use any kind of stressful situation. Psychologists tend to use shock. Now, shock is such a traumatic event. You have all kinds of stress components entering into the learning experience. It's going to hide the learning experience. You may be getting changes that are actually a function of stress. They have nothing to do with learning per se. Shock is very traumatic, increases the levels of hormones, increases general neural activity. So this is not a preferred way of doing things. Often you have surgical techniques where you implant an electrode or you implant a micro cannula to inject materials. The surgical shock is again traumatic and there's stress. So there's no stress, there's no surgical stress, there's no mechanical stress, and there's no shock. So this is a very clean manipulation which would give you an ideal opportunity to look at changes that are specific to the learning experience. Okay, so once he's got these animals fairly well trained, on day 5 now, where they're still learning the experience, he injects a micro quantity of a labeled amino acid. Now, he injects 60 microcuries. Okay, a curie is a measurement of the amount of radioactivity. Everyone knows that radioactivity is the emission of some kind of particle or some kind of radiation. In this case, he used tritiated lysine. Now, tritium is basically hydrogen with 2 extra neutrons. And it's unstable, and it emits a high-energy beta particle when it decomposes, and it becomes helium. Okay, this beta particle is an electron. It emits an electron. So he injects this lysine now 15 minutes after training. No, wait. He injects a half an hour before training, the final training period. He injects it. And 15 minutes after training, he sacrifices the animal. He teases out single pyramidal cells from the hippocampus. Right. Okay, a half an hour before the final training period. The training period lasts 25 minutes. Pardon me? Hippocampus. Okay, now 15 minutes after training, he sacrifices the animal. He teases out these individual hippocampal cells and looks for the proteins that have incorporated this labeled lysine. He teases out roughly 300 of these cells, and then he would homogenize them. And then, like Rick talked about, you go through a procedure where you purify this and you apply it to a gel. If you remember from Rick's talk about electrophoresis, you create a gel. And this gel, you can think of a gel as nothing more than a sieve with various size holes, which get continuously smaller. So this is a schematic representation of the holes in a gel. Proteins are drawn because all proteins have a charge associated with them because of their acidic and basic amino acids. They have a charge and they're drawn down until they're stuck and the hole is no longer large enough to permit them to go any further. So what you have is a distribution where you have bands created everywhere where the proteins of a particular size get stuck. So the lighter ones go all the way down, and the big ones stay up near the top. Now he found out that in general, when he compared the protein synthesis activity of trained versus control or untrained animals. But there was largely no difference between most of the bands, which is to be expected. It was actually kind of nice. But the two bands, which he called fraction 4 and 5, that were somewhere in the range of 30,000 molecular weight, 30,000 Daltons. The bands 4 and 5 seem to increase with training. Okay, so he took groups at day 5 and then at day 7 where they really seemed pretty much almost fully acquired the task by then. And day 10, all right, where the task is really pretty well acquired. There doesn't seem to be any more learning acquisition. And his data looks like this. Group 1, in which you had 5 animals. This is such a hard and difficult task that you find you don't have very large numbers. In the rest of psychology, you can run 100 subjects, 200 subjects. But when you get into biochemistry, you really end up with very small numbers, which is one of the problems, because any small deviation that just happens to occur may give you an artificial difference when you don't really have one. So that's something you have to be aware of when you look at this literature. Okay, so he takes 5 of these animals, he runs them, and then he finds out that the activity associated with fractions 4 and 5 is 3.3 CPM, which is counts per minute, that is, per microgram of protein. That means that in 1 microgram, which is 10 to the minus 6, 1 microgram of this protein, he can count 3 decays. That means that 3 hydrogen, or 3 of these tritiated lysines have decayed. Okay, so this is just a count then proportional to the amount of protein that's there. It has lysine in it. So this is like saying there's a certain amount of this lysine incorporated. Now, in group 2, which is at day 7, we found roughly the same thing, about 3.9. Okay, in group 2A, which I'll get back to in a moment. And then there's group 3. Group 3, which is at day 10, they found out that only about 1.8 CPM per microgram protein were recorded. In other words, roughly only half the amount of protein in this fraction is around, because it's a direct proportion to counts per minute and the amount of protein that's been synthesized. At least that's roughly the assumption. And in the controls, which had no training at all, were 1.5, a little less than half. Now this is really nice. Essentially, what this is saying is that in these hippocampal cells, increased protein synthesis is correlated with the learning acquisition phase. But after learning has actually occurred, you no longer see this. In other words, now he's just performing. He's not really learning anything. And it goes back down to almost a baseline control level. Now, another thing that he did was he thought perhaps he's training them too slowly, giving them too much time, and he's kind of diffusing out the learning experience. So he gave them much less training. He gave them like 1 training experience a day instead of 2 for a shorter period of time. That was his group 2A. We found that under these circumstances, he got an enormous increase in the amount of proteins in fractions 4 and 5. Okay, now he also looked way down here at the bottom of the gel. And he found out that there was 2 faint bands in, this is a well, okay? This represents 1 animal. That in trained animals, there was 2 faint bands present, and in untrained, was only one. So he started looking at this, and he found out that if you created the antibody to S100, now this is a complicated procedure, but it's very brief. What happens is you can create antibodies to any kind of protein or foreign invading substance that enters the body. And this is the body's natural defense mechanism to prevent disease. Because you get measles, that you create antibodies that are against specific proteins that are on the invading infectious organism, and that the next time you get exposed to measles, the antibodies come and they clump around and they destroy that infectious agent. So you can use antibodies in science to become specific labels for any specific protein. What you do is you isolate this S100 fraction and you inject it into rabbits, is what they tend to do. And then they cultivate these rabbits with a number of injections, and then finally they bleed the rabbit and they get out the antibody fraction and they clean it up, and then they have an antibody which, whenever S100 is around, it binds to it. Okay, now they can tag the antibody with a radioactive tag or with a fluorescent tag, so that wherever the antibody sticks, you can see it, or you can observe it somehow. So we found out that if you applied antibodies to S100, that they stuck, both of these bands. Okay? So he's saying, okay, this must be that S100 protein that other people had described previously. But again, this is an S100 that's found inside cells, the nerve cells, near the nucleus, or in the nucleus of these cells. And he also found out that this band right here, the second band present in the trained group, would come out of the gel when you use saturated ammonium sulfate. So with this test, an antibody test, he says that this is the S100 fraction, and it seems to change in response to training. But they're both kind of S100s, but one is the S100 that has been observed earlier. Okay, to confirm this idea that the S100 is a training unique protein, or at least the second S100 is unique to the training condition, he used the same handedness task, and this time in the training of these animals, when they were learning to pull the pellets out, that on day 3 of the learning task, he'd inject this antibody against S100 into the ventricles of the brain. And then as a control, he'd injected another antibody that was not specific for S100, but was just a blood antibody. And that he found that training was not really acquired. And that for both groups, okay, the open circles will represent the trained group with S100 antibody injections. And then I have closed circles representing the controls, which are injected with another antibody that has nothing to do with S100. Okay, the injection causes both of them to have a decrement in learning. But that following this, the control groups kind of pick up and continue in basically an extrapolated fashion of the way they should have. Whereas these that have been injected with antibodies of S100 never really seem to acquire the task much more. So he's saying this S100 is very crucial to the acquisition of performance.
The control groups were given antibodies not specific to any protein?
Well, there's a general gamma globulin, okay? I think it's specific to an irrelevant protein, or a certain type of protein. So this is some of the strongest evidence which suggests that these proteins are intimately involved with the learning process and long-term memory. because you don't see any kind of effect after 24 hours of the learning. Okay, now, who has the time? I just want to know how much more time. 5 minutes. Have 5 more minutes? Okay. I guess what I'd like to do now is kind of give you a model of perhaps what these proteins are doing, give you a sense or a feeling for what may be happening. Again, this is only a model. It's a very crude one. You might have an idea of what proteins are doing, and then I'll refer back to this protein turnover and try to give you an idea of why it may be turning over, but we may still have long-term memory. Yeah.
Just playing the devil's advocate for a second, would it not, at least at that time, been thought possible that, okay, rather than function in memory, these still function in perhaps building on the special structures to the cell that cause reconnection of the cells to the areas, he was talking about reverberation. So might not these proteins be the ones specific for adding onto the cell and causing new connections?
Yeah, but you see, that may be memory.
Okay.
In fact, essentially this is the model I'm going to propose. Now, I told you earlier that any nerve cell can communicate with any other nerve cell in the central nervous system within about 5 synapses. Okay, so you have this enormous connectivity. Anything can connect to anything. This is very nice. You have your brain, okay, make a rat brain, we're getting used to it. Focus closer. Now, let's say that we have a simple task. And again, it's very simplified, so you can't get real picky, but try to understand. We have a task that's going to be learned. The animal is going to dip in and get a pellet out. Okay, so he has visual cues, and he's got olfactory cues. Okay, so he's got stimulation of these two senses. Okay, and what he wants to come out is some kind of motor behavior to dip his hand in and get a pellet out. Okay, so here is kind of like the target. Okay, so let's just take the visual cues and make it a real simple model. There are ways by which this cell can communicate with this cell. And the training, let's say, is the increase of connectivity between this cell and this cell. Okay, connectivity, someone asked about yesterday. That is, the connections are somehow enhanced. So if we get down to a synaptic level now, we could say that if this is the synapse connecting that nerve, that particular neuron to the other neuron, or maybe there's 5 of them, but here's one. And here we have the neural transmitter. Here we have the postsynaptic dendrite. And we have presynaptic. But a number of things can happen to change the connectivity. For example, you can have more neurotransmitter, okay? That you can put more neurotransmitter in every one of the vesicles. So protein synthesis could perhaps be more enzyme that grabs neurotransmitter, synthesizes neurotransmitter, and puts it in the vesicles, okay? That would be one way. So that each time you release an action potential release vesicles, more would get into the synaptic cleft. Okay, at the same time, postsynaptically, you have receptors, okay? These receptors are sitting on the postsynaptic side, and you could increase the number of receptors so that the same amount of neurotransmitter could cause a bigger effect, a larger EPSP or a smaller EPSP, but a bigger effect on the postsynaptic membrane. Another thing that could happen is there's a cleft material that's proteinaceous that holds the synaptic cleft and the two membranes in very close apposition. This cleft material can perhaps become more efficient, can bring the cleft closer together. So this is just a few mechanisms by which you can change synaptic efficiency. And essentially, what I would like to postulate is that you have ongoing neural activity where by training you have a temporary increase in connectivity. And I refer back to short-term memory and to phosphorylation. Let's say that phosphorylation causes more of these receptors to work. Okay? Phosphorylation causes more enzyme activity, increase in tyrosine hydroxylase. I think you mentioned that's one of the effects of cyclic AMP. Okay, now, at the same time, the cyclic AMP or some other messenger would go back to the cell nucleus and say, okay, make this a permanent effect. The cell would hold on in short-term memory, which lasts from about 15 seconds to an hour. And that would hold the efficacy at a higher level, and then protein synthesis would come out and kind of cement it in a more permanent fashion. So we have this enormous amount of protein synthetic activity, and this would be support to learning function. That you need this, it's an ongoing process because every organism in a mammalian system, for certain, where there's a lot of learning going on, it's necessary for survival, for him to be able to learn, to be able to change his behavior, to be able to adapt, and be able to figure out new things. So this thing has to go on all the time. At the same time now, I pointed out that this protein is turning over all the time. It's never around very long. Now this is kind of almost contradictory with the concept of long-term memory. But I think that there's two things that may help to understand why this happens. One, the proteins wear out. They only last a certain length of time, and then they denature, or they get degraded, or something falls off, or they get lost, okay? Now, with this continual loss, any kind of permanent connectivity change you're going to have will fade. So what you really need is, it's almost as if the brain is not taking any chances of losing its memory. That it's going to remake that protein every 8 hours, every 14 hours, every 8 days. It's going to make it over and say, well, I don't really know if that protein is still good or not. To anthropomorphize it. I don't know if it's good or not, but I'm going to throw it out and put a new one that I know is good there. I'm just going to keep on doing this to maintain it so I can be absolutely certain that this is recorded correctly. So we have this ongoing metabolic activity to ensure that these connectivity changes, these memories, are permanently recorded. That's it. Yeah.
On that graph, was there an increase past day 14, or was there some sort of change in the shape of the graph at day 14?
It would peak out. Although they didn't talk about it. Yeah.
What about memory like recall memory? With human
Retrieval. That was kind of one thing I wanted to talk about, but I really don't have the time. We'll probably get into it tomorrow, either with me or Dr. Rupert, depending on who's here. But retrieval now is a fully understood process, and may be electrical, okay? But really for the storage of the memory, it seems that it has to be somehow metabolic. Thank you.